Welcome. Welcome. Today we have one of our own faculty members, Larry Zweifel, who's a PhD doctor, to speak with us, and he's going to tell us about the genesis of his talk and where it comes from. But let me introduce Larry. He describes that his research is to investigate the neural circuitry of emotional processing through the implantation of techniques in mouse behavioral genetics. He began training as a research associate at the Johns Hopkins University School of Medicine in the laboratory of Dr. Gary Wand. He investigated the role of 3,5-cyclic adenosine monophosphate signaling in the sedative hypnotic effects of ethanol and ethanol consumption, utilizing mouse genetic approaches. He did graduate studies at Hopkins University School of Medicine, Department of Neuroscience. His thesis work was in the laboratory of Dr. David Ginty, and his PhD dissertation focused on the mechanism and function of retrograde neurotrophic factor signaling in sympathetic nervous system development. Following graduate school, he trained as a postdoctoral fellow at the University of Washington, Department of Biochemistry, in the laboratory of Dr. Richard Palmetier. Utilizing mouse behavioral genetic models, he investigated the genetic regulation of dopamine neuron activity as it relates to substance abuse, learning, fear, and anxiety. Through an NIH grant, he became assistant professor of pharmacology and psychiatry at the University of Washington. He now investigates the contribution of N-methyl-D-aspartate type glutamine receptor and the small conductance calcium-activated potassium channel, SK3, in the control of dopamine neuron firing patterns in response to rewarding and aversive information, and the contributions of these firing patterns to the activity of forebrain structures involved in fear and reward processing. He has discovered that NMDAR-dependent burst firing by dopamine neurons is essential for both fear and reward processing. Thus, current investigations are aimed at understanding the basic mechanisms by which dopamine neuron burst firing regulate the neuron circuitry of emotion. So, thank you very much, Dr. Zweifel, for speaking with us.
Thank you, Sharon, for the invitation. Can everyone hear me okay? Mm-hmm. All right. So thanks again, Sharon, for the opportunity to come down and interact with some of my new colleagues in the Department of Psychiatry. So as she mentioned, I'm going to be discussing the work that I've been doing the last several years and now have started in my own lab, dissecting the role of dopamine in fear and anxiety processing. So as most of you are aware, dopamine is a modulatory neurotransmitter that is synthesized by a population of neurons located in the ventral midbrain. These neurons send dense projections to numerous limbic structures, such as the prefrontal cortex, the amygdala, the hippocampus, as well as the dorsal and ventral striatum. It's not surprising, based on the targets of dopamine neurons innervating limbic circuits, that this neurotransmitter system may play an important role in both motivation, goal-directed behavior, as well as emotional processing. Also, not surprisingly, as most of you are aware, disruptions of dopamine signaling is associated with numerous neurological and psychiatric disorders, most notably Parkinson's disease, schizophrenia, and addiction, but it's also been implicated in attention deficit hyperactivity disorder, as well as anxiety, depression, and obsessive compulsive disorder. So what we're interested in unraveling is how this neurotransmitter, dopamine, regulates such a diversity of behaviors. And we think that we can gain some insight into how dopamine may be doing these various functions based on the different types of activity patterns that these neurons display. So in an awake, freely moving animal, or humans, there are three main patterns of dopamine neuron activity. There's a bursty pattern of activity in which you have action potentials, which are fired at a irregular interval, and you have brief epochs of activity where you have spikes of action potentials that occur with a relatively high frequency. This causes a high level of neurotransmitter release in the synaptic terminal, which is referred to as phasic dopamine release. You also have a pattern of activity which is referred to as pacemaker activity, so the cell's firing at a relatively constant rate with a relatively constant interspike interval, and this is thought to be associated with the slow, steady release of neurotransmitter in the terminal field, and it's referred to as tonic dopamine release. And finally, you can have, of course, inhibitions of dopamine neuron activity, and these inhibitions or pauses in activity are thought to result in transient pauses or dips in neurotransmitter release, which can relieve the activation of specific types of neurotransmitter receptors, which are thought to be tuned to tonic activity, which are normally inhibitory, which would disinhibit those cells and allow them to engage in circuit function. So there are, as you're probably also aware, two main families of dopamine receptors. There's the D2 receptor family, which is an inhibitory heterotrimeric G protein complex that couples to the G protein G-alpha I. So activation of D2 receptors inhibits the formation of cyclic AMP mediated by adenylate cyclase, and also activates G protein coupled inwardly rectifying potassium channels. The function of this receptor overall is to dampen or suppress the activity of the cell. Now, the D1 family members are coupled to the stimulatory heterotrimeric G protein complex, G-alpha S. These ion channels stimulate the formation of cyclic AMP, initiate downstream signaling events, such as the activation of pKa, and can modulate ion channels in the membrane positively, which overall serves to facilitate or increase the excitability of a cell. Now, these two receptors are thought to be selectively tuned to distinct patterns of dopamine neuron activity based on their relative affinity for the neurotransmitter. So the dopamine D2 receptor has a relatively high affinity for dopamine, and it's thought that this high affinity for dopamine is what allows this receptor to be selectively activated by low levels of tonic dopamine release into the terminal fields. So if you have a single action potential, you get a little bit of dopamine release. This little bit of dopamine release is sufficient to engage the D2 receptor and keep those cells which are expressing D2 receptors in a relatively quiescent state. Conversely, if you have a burst of action potentials, you get a synergistic increase in the amount of dopamine release. This high level of dopamine release in the synaptic cleft is thought to preferentially activate the D1 type dopamine receptor, which has a relatively low affinity for the neurotransmitter. So what we're interested in is trying to figure out if we can use genetic tools to try to disrupt specific patterns of dopamine neuron activity. And we're specifically interested in disrupting this phasic pattern of activity. There are four main ion channels in dopamine neurons which have been implicated in the regulation of the firing patterns of these cells. That is the GABA A receptor, which is, you're probably familiar, is the principal inhibitory neurotransmitter in the brain. So it's thought that you require a suppression of GABA release, a disinhibition of the cell through the inactivation of the GABA receptor, which allows the cell to be excitable. You have neurotransmitter receptors, the nicotinic acetylcholine receptor, as well as the glutamate receptor, the NMDA type glutamate receptor, which are thought to actually work synergistically to facilitate phasic activation of the cell through their ability to flux calcium into the cell. And finally, you have an ion channel called the SK3 channel, which is a slow and activating calcium-sensitive potassium channel. So calcium influx through the acetylcholine receptor, as well as the glutamate receptor, facilitate activation of the SK channel, which allows potassium efflux, which hyperpolarizes the cell and terminates the burst of action potentials. So our first approach has been to selectively target the NMDA type glutamate receptor explicitly in dopamine neurons and see how this affects patterns of activity and animal behavior. To do this, we use a multi-disciplinary approach, which involves mouse genetics, in vivo electrophysiological recordings from mice, as well as mouse behavior. So we want to use what we know about mouse genetics to selectively inactivate genes in specific populations of neurons, see how this alters the firing properties of these cells, and what the correlation is between these specific patterns of activity and the ultimate outcome, which is animal behavior. So to do this, we utilize an approach which is referred to as conditional gene inactivation. So we have the allele for the essential subunit of the NMDA receptor, NR1, which is encoded by the GRIN1 gene, which is flanked by DNA recombinase sequences that are recognized by a specific type of recombinase called circular recombinase or Cre recombinase. So in the presence of Cre recombinase, it facilitates the recombination between these two DNA sequences and will remove a major coding region of the gene, thus rendering it inactive. We can express this Cre recombinase through the endogenous dopamine transporter gene, which is selectively expressed in dopamine-producing cells. So by crossing these animals, which have the SLAC6A3 Cre allele, which encodes the dopamine transporter, and the GRIN1 LOX allele, we can selectively inactivate the NR1 subunit and dopamine neurons. And this is an example of an electrophysiological trace from midbrain dopamine neuron, where you can see the glutamate responses. You have an AMPA receptor component and an NMDA receptor component, which are the two principal ion channels that mediate glutamate receptor signaling. In the absence of the NR1 subunit, we have no NMDA receptor component to the excitatory postsynaptic current. And in non-dopamine neurons in this animal, the NMDA receptor component is intact. So we have, in fact, selectively inactivated this ion channel in dopamine neurons. So the next question is, okay, we've inactivated this ion channel, which is thought to be important for burst firing. What actually happens to the activity of the cells? So to investigate this, we do in vivo electrophysiological recordings from freely moving mice. So essentially what we have is a device which is implanted chronically in the animal. This is a drivable tetrode system. So you have 4 wires which are wrapped together, which are lowered into the brain to a specific brain region. We can then detect the electrical signal in the brain and amplify it and see how activity patterns change over time. And this is what a typical trace looks like from an animal that's being recorded. So you have multiple cells that you're detecting. You can isolate these cells based on their relative position to the tetrode and plot their energies relative to each other, and it will give you a scatter plot, such as this, which then allows you to isolate individual units or individual cells. So how we screen for dopamine neurons is we look for cells which have a firing pattern typically less than 10 hertz and are responsive to the D2 selective agonist quinpirole. So dopamine neurons have D2 autoreceptors on them. When D2 receptor agonist is injected, you get rapid and prolonged inhibition of these cells. And this can be reversed by the D2 selective antagonist aticapride. So we're looking for cells that have a specific firing rate, have a specific action potential waveform, and are sensitive to quinpirole. Once we've identified these cells, now we want to see if we can look for alterations in activity patterns. And what we're looking for specifically are these activity patterns which are referred to as burst epochs. These epochs of activity have been well characterized for about 30 years now, starting with the work of Anthony Grace, in which he showed that a burst of action potentials can be characterized by 2 spikes that occur with a relatively short interspike interval and is terminated by a subsequent spike that has double the interspike interval between the first 2. So we're looking for these patterns of activity in these animals and seeing if there's a difference between the control animals and the animals that lack the essential subunit of the NMDA receptor. And what we see is consistent with what we had predicted. There's a significant reduction in the overall burst firing of these animals as analyzed by burst set rate, the number of spikes per burst, and the duration of the burst of action potentials. So now we can ask the question of whether or not this burst firing of dopamine neurons does in fact facilitate transmitter release into the terminal fields. So it's been proposed that when you have multiple action potentials occurring with a relatively short interspike interval, that this causes a synergistic increase in calcium influx into the nerve terminal. This influx of calcium into the nerve terminal allows a greater amount of synaptic vesicles to fuse to the membrane, leading to an overall increase in neurotransmitter release into the terminal field. So if we have disrupted the ability of the cell to fire action potentials with a relatively short interspike interval, we should have disrupted the ability of the cell to have phasic amount of transient high concentration of neurotransmitter release. So to investigate this, We collaborated with Dr. Paul Phillips here at the University of Washington using cyclic voltammetry, which allows him to measure quantiles of dopamine release, both in awake animals as well as in anesthetized animals. And in this experiment, essentially what we're doing is we're taking an anesthetized mouse and we're lowering a stimulating electrode into a region that innervates the dopamine neurons, stimulating this brain region, and then measuring dopamine release in the striatum, a dopamine terminal field. And when you increase the intensity of the stimulus into the area innervating the dopamine neurons, you get a proportional increase in the amount of dopamine that's being released. So now we can look at this stimulus intensity response curve in normal animals and in animals that lack this NMDA receptor and see if we have in fact disrupted phasic dopamine neuron release. And that is indeed what we see. So you can see in the control animals, as you increase the stimulation intensity, you increase the amount of dopamine release. In the knockout animals, you reach a plateau that is much earlier than that observed in the control animals, and they never reach the level of neurotransmitter release that is comparable to that of the control animals. So we have, in fact, not only disrupted the ability of these cells to fire burst of action potentials, we've disrupted the ability of these cells to have transient high concentrations of transmitter release into the terminal fields. So now what we're interested in is determining how this pattern of activity is important for emotional processing. And what we're interested in is negative emotional valence, or how this neurotransmitter system may be involved in processing aversive or fearful outcomes. It's been demonstrated numerous times, but this is the most recent paper identifying the role of dopamine in aversive processing. And this is single unit recordings from an anesthetized animal where they're delivering a foot shock, which, as most of you are aware, if you repeatedly deliver a mild foot shock to an animal, it will avoid areas where that foot shock was delivered and learn to associate cues that predict that aversive outcome. So in dopamine neurons, when you deliver a foot shock, you have two distinct patterns of activity. You have a number of dopamine neurons which are transiently inhibited, but you also have a number of dopamine neurons which are transiently activated. And what we want to know is if we disrupt these cells which are transiently activated by the aversive stimulus, how does this affect aversive stimulus processing? So going back to our in vivo electrophysiological recordings in the mice, we do in fact see multiple modes of dopamine neuron responses to aversive stimuli. We see both cells that are inhibited by the aversive outcome, cells that are stimulated by the aversive outcome, and cells that could really care less whether the aversive outcome occurred at all. So, and there appears to be no discernible features between the dopamine neurons that would allow one to a priori determine whether or not this cell would be inhibited, activated, or show no change in activity. However, the firing patterns, again, are significantly attenuated in the animals that lack the NMDA receptor. And then when we look at the population activity from these animals following an aversive experience, so this is the tail pinch. So we're recording 10 sessions that are about a minute each, and halfway through the session, we give the animals a 5-second tail pinch, and we look for changes in activity. And this is a control animal. You can see the heat plot of the activity. There's a robust induction of activity in a specific population of dopamine neurons with this aversive stimulus. And in the knockout animals, it's significantly attenuated. When we average these across animals, we see, consistent with the 3D data, we see a significant attenuation of the amplitude of the response to the aversive stimulus. However, the response of the cells that are inhibited by the aversive stimulus appears relatively unchanged by the absence of the NMDA receptor. So now we can ask specifically, if we disrupt the activation of the cell but not the inhibition of the other cells, what happens to fear processing? So how do we study fear processing in the lab? To do this, we use a technique which is referred to as fear potentiation of the acoustic startle reflex. So in this, you take a mouse and you put it into this recording chamber that has a small shock grid on the floor. And you play a loud blast of noise and measure how high the animal jumps. So underneath this holding chamber is a piezoelectric accelerometer. So when the animal jumps, it converts that force into an electrical signal, and we can monitor the amplitude of that electrical signal, which is proportional to the amount at which the animal is jumping. Now, to condition the animals, we put them in the chamber. We show them a light. For about 10 seconds, the light goes off, they get a foot shock. They get this pairing anywhere from 10 to 30 times. We then put the animals back in the chamber. We show them the light, And now, instead of giving them the foot shock, we give them a loud blast of noise. And what happens when you do that is you can see, if you look at the startle amplitude of an animal that's been conditioned, in the absence of the cue, the animal will jump with a specific force relative to the intensity of the acoustic stimulus. However, if there is a cue that's been displayed that is associated with a negative outcome, the animal will jump significantly higher than it would in the absence of the cue. And this is referred to as fear-induced potentiation of the acoustic startle reflex. So in control animals, as I demonstrated, you can get a nice, robust induction of fear-potentiated startle. And then when we looked at the knockout animals, what we saw was rather surprising to us. We saw a disruption of the cue-selective increase in fear-potentiated startle. But we saw an increase in the acoustic startle reflex, both in the presence and in the absence of the cue. So when you score this as a fear-potentiated startle, which is the canonical way of displaying this data, you would look at it and you would say, okay, well, these animals learn nothing. But that's not entirely true. If you look at the raw data of the startle amplitude, these animals appear to have made some association, or at least appear to have some alteration in the acoustic startle response after fear conditioning. So the question is, do these animals have impairments in cue-selective fear conditioning? And they're using an alternative approach, which would be potentially a context-dependent association, which allows them to associate the cue. So this is the same context that they're getting the acoustic startle in that they received the foot shock in. So it's possible that these animals have formed some contextual association, which is resulting in the manifestation of the increased acoustic startle amplitude. So we can do that after 24 hours, and we can also see this after 10 minutes. So you can do the same paradigm in which you shock the animal and then pair with a cue and see if they can get an increase in fear-potentiated startle. Now, in control animals with this paradigm, you also see an increase in the acoustic startle amplitude in the absence of the cue. And this is referred to as shock sensitization. So after an animal has been repeatedly shocked, they are in a heightened state of fear. And then when you measure their acoustic startle reflex right after that, they are still in this elevated state of stress and fear, and they will have a generalized potentiation of the acoustic startle reflex. But the knockout animals still have significantly elevated acoustic startle responses compared to the control animals. And again, there's no cue-elicited increase in the fear-potentiated startle. So the question is this potentiation of the acoustic startle reflex context-dependent? Are these animals using an alternative learning mechanism in the absence of their capacity to learn specific cue associations? So to address this question, what we do is we put the animal in the acoustic startle chamber. We monitor their acoustic startle response to multiple startle pulse intensities. We then take them and the next day we give them 10-foot shock pairings in a novel context. In a different lab, different investigators, different shock grid, everything is completely different than the context in which they received the acoustic startle reflex. They go back to their home cage for either 10 minutes, 24 hours, or a week, and we monitor their acoustic startle response again. And what we found was these animals manifest an increase in the acoustic startle response irregardless of the context in which the shock was given. And it occurs at 10 minutes, 24 hours, and 1 week after shock. And this was really interesting to me because sensitization of the acoustic startle response is a hallmark feature of some psychiatric disorders, including post-traumatic stress disorder. So these animals may be manifesting a PTSD-like behavioral phenotype after an aversive experience due to their inability to appropriately encode the specific information about the outcome. So, is this potentiation of the acoustic startle reflex associated with other forms of generalized fear or anxiety? So to monitor this, we use two standard methods to investigate anxiety in laboratory animals. The first is an elevated plus maze. So for those of you who aren't familiar, it's a maze that sits about 3 feet off the floor. 2 arms of the maze are enclosed, 2 arms of the maze are open. Mice are generally anxious, so they don't explore the open arms of the maze as much as the closed arms, but they will go out and explore those open arms to some degree. So we can look for changes in this willingness of an animal to go out and explore what would be considered an anxiogenic environment. And what we see is during baseline conditions, there's no significant difference between the knockout animals and control animals that are either going to receive foot shock or control animals that are not going to be receiving foot shock but be repeatedly exposed to the environment. Now we shock the animals and we measure their elevated plus maze and open field activity again, and we see a significant reduction in exploration of the open arm in the knockout animals. And when we look in the open field, the willingness of the animal to cross through the center of the open field is also drastically reduced in these animals following aversive conditioning. So this would suggest that after an aversive experience, these animals are manifesting a generalized fear and anxiety-like phenotype. We looked at other aspects of fear and anxiety and stress in these animals. We looked at pre-pulse inhibition, which is a measure in both humans and animals of disrupted cortical function and sensory motor gating. These animals have no alterations in pre-pulse inhibition either before or after the aversive experience. Their corticosterone levels are normal relative to control animals before and after foot shock conditioning. And their level of monoamine neurotransmitters and monoamine neurotransmitter metabolites, dopamine, norepinephrine, and serotonin, are all normal pre and post conditioning. So whatever the alteration is in these animals, it is independent of alterations in the HPA stress axis or alterations in the overall level of monoamine neurotransmitters or monoamine neurotransmitter metabolites. So the next question is, which population of dopamine neurons is responsible for mediating this particular phenotype? So there are multiple dopaminergic nuclei in the brain. There's the midbrain dopamine neurons, which I've discussed. There's also a population of dopamine neurons in the periaqueductal gray region of the brain, which is important for processing nociceptive information. So these animals may have disruptions in their ability to sense pain, which could be altering their behavioral responses. There's dopamine neurons in the olfactory bulb as well as the retina, which could disrupt these animals to interact with their environment, which could be leading to their alteration in behavior. And there's also dopamine neurons located in the hypothalamus, which could be disrupting hypothalamic function and subsequently altering the abilities of these animals to cope with stressful situations. So to address this question, we utilize a technique which allows us to selectively reintroduce the gene that we've inactivated exclusively in the cells in which we originally inactivated the gene. So this is a conditional viral approach. So we have a virus which we can inject into the brain, in which the expression of the NR1 subunit is prevented by a transcriptional translational stop cassette that's inserted into the open reading frame of the gene. Now, this is flanked by Cre recombinase recognition sequences. So the Cre recombinase that we use to initially inactivate our gene can now turn our gene back on. So we can turn it back on only in the cells that we took it out of. And we can do this with some anatomic specificity because we're injecting it only into a specific brain region that controls a specific dopaminergic population. In this case, we're injecting it into the ventral midbrain. And as you can see here, we've labeled this NR1 subunit with a specific epitope that allows us to detect it with an antibody. And we can see a nice overlap of our NR1 expression in our TH positive cells. Of course, TH is tyrosine hydroxylase, which is the rate-limiting enzyme in dopamine synthesis and is selective for dopamine neurons in this region. We can look at the electrophysiological patterns in these cells, and we see a restoration of the NMDA receptor component in the dopamine neurons of the virally rescued animals. We now take these animals that have been virally rescued, we give them a foot shock, and we monitor their acoustic startle reflex and their anxiety measures and elevated plus maze and open field, and we see that reintroduction of the NR1 subunit specifically into the ventral tegmental area prevents the manifestation of the behaviors that we observed in the original knockout animals. So what are these animals telling us about fear and anxiety processing? So the current hypothesis, which is based not only on my work but the work of many other labs, is that the distinct patterns of dopamine neuron activity that are observed during aversive outcome are a reflection of distinct roles of this neurotransmitter system encoding different aspects of emotion, both salience, or the importance of an event, and the valence of an event. So during aversive or fearful outcomes, you have a population of dopamine neurons that are normally activated by this stimulus, which are telling forebrain structures that something important happened that was unexpected. You also have a population of cells which are communicating with other brain regions, which are transiently inhibited. These cells are telling that brain region that something of negative valence occurred. So if you have salience detection and negative valence detection simultaneously, that facilitates conditioned fear. Now, in animals in which you've disrupted their ability to be activated by unexpected stimuli or salience detection, the animals cannot appropriately encode the events surrounding the aversive outcome, but they know that something negative happened. So when you have a aversive outcome with the inability to encode what the aversive outcome is, you get a generalization which manifests as anxiety and generalized fear. Conversely, in these animals, we've looked at numerous appetitive conditioning paradigms. And in this situation, what you would have for an appetitive or rewarding experience would be activation, again, of the salience detection system, as well as an activation of the valence detection system because you have something of positive valence or reward. When you have salience detection and positive valence, you get reinforced behavior. Now, in these animals, what we see, we have an attenuation of both the salience response and the valence response, and these animals have delays in reinforcement learning as well as impaired performance. So now that we've sort of identified that midbrain dopamine neurons appear to be important for processing aversive information and anxiety, what we want to know is what are these populations of cells that are activated by these stimuli? Where do they project? And if we can define their projection targets, what does that tell us about how dopamine is involved in processing this information? So in order to do this, we're currently implementing a new technique in the lab, which is referred to as fiber-optic fluorescence microscopy. What this allows us to do is to image fluorescence and deep brain structures of freely moving animals. We can couple this with the detection of fluorescence that is responsive to specific patterns of activity. So we can encode these detectors using genetic tools so we can specifically identify patterns of activity in specific populations of cells. So how this works is you have a probe, which is about 300 microns in diameter, which consists of approximately 3,000 1-micron fibers that are bundled together. So you have an excitatory source, which is a laser, which passes light through the probe into the brain, which excites tissue expressing genetically encoded fluorescence indicators. These molecules fluoresce in response to the light stimulus. That light that is emitted by these fluorescent proteins is then detected by the probe and sent back to a photodetector, which allows us to actually visualize proteins in the brain. So this is what it looks like, imaging various localized fluorescent proteins in the brain. So we can see proteins that are localized specifically to the nucleus. So this is a nuclear localized GFP. We can isolate fluorescent proteins which are expressed exclusively in the cell bodies. And we can also localize both green and red fluorescent proteins. So what we're intending to do is couple this technology with the capacity to monitor patterns of neural activity. So how do we detect patterns of activity using genetically encoded fluorescent indicators? We do this utilizing a protein which is referred to as GCAMP3. So this is a protein in which you take a green fluorescent protein, which normally fluoresces. You cut it in half and invert the N terminus and the C terminus. And this makes it not fluoresce very well. So you can put a calcium sensor, calmodulin, on this new protein, as well as a protein that interacts with this calcium sensor when calcium is bound. So when calcium binds, it causes a transformation of this protein, which makes it fluoresce again. So it goes from relatively unfluorescent to a high level of fluorescence. And this allows us to detect changes in intracellular calcium, which are dependent upon the activity of the cell. So when a cell fires, you get an increase in calcium flux into the cell. This calcium flux will bind to the genetically encoded calcium indicator, give an increase in fluorescence intensity, and we can monitor the magnitude of activity in the cell. So just as a proof of principle. We've generated a construct which allows us to conditionally express this fluorescent indicator. This is cells in vitro, so you can see some fluorescence in these cells in the absence of stimulation. And then when we stimulate these cells with the calcium ionophore, were you guys able to see that? There might be too much light in here. Let's try it again. Yeah, it's a lot prettier than that on my screen, unfortunately. But so we can stimulate an increase in fluorescence intensity of this molecule in cells in culture. And this is just a temporal response of the increase in fluorescence intensity after the application of this ionophore, which causes calcium to flux into the cell. So we can then inject this, a virus encoding this fluorescent indicator into the brain, and we can look for changes in activity in freely behaving animals. So this is an animal that's been injected with a virus that encodes this genetically encoded calcium indicator. And now we're giving this animal a tail pinch, and we're looking for changes in fluorescence intensity in dopamine neurons. So as you're going along, then you give the animal a tail pinch, and you can see the yellow and the red is the increase in fluorescence intensity. This is basically just showing a heat plot of the data. So the red is the highest level of intensity. So when we give the animals a pinch on the tail for 5 seconds, we see this increase in calcium flux into the cell, which is similar to what we see electrophysiologically when we record the activity of these cells. So now what we can do is actually utilize this virus in combination with viruses that allow us to retrogradely label cells so that we can express this genetically encoded calcium indicator and dopamine neurons that project to specific targets, so that we can map those cells which are activated by the aversive stimulus and develop a functional map of dopamine for aversive processing. So the last thing I want to talk about in the last few minutes is our work studying the role of the D1 receptor. As I mentioned early on, the D1 dopamine receptor is thought to be selectively tuned to this phasic pattern of dopamine neuron activity. So what we want to do is map the areas where dopamine neurons that are activated by aversive stimuli project, and then determine whether or not this receptor is in fact responsible for mediating those effects of that transmitter release. So to do this, we're utilizing another genetic tool. So we want to isolate the specific targets of dopamine neurons, be it the prefrontal cortex or the amygdala or the striatum or the hippocampus. They're minimally essential for mediating dopamine-dependent fear processing. So to do this, we're utilizing an animal in which the expression of the endogenous D1 receptor gene is disrupted by the insertion of this Cre recombinase into the open reading frame of the gene. So an animal that is homozygous for this Cre is essentially a D1 receptor null animal, so they don't express any D1 receptors anywhere in the brain. We now have a virus that allows us to inject a construct specifically into the specific brain regions that allows us to turn the D1 receptor back on. So we can reintroduce D1 receptor signaling only in specific populations of cells and determine whether or not signaling in those cells is the minimal essential requirement for mediating aversive processing. So just to give you a background of the D1 receptor knockout animals, when we put them through the aversive conditioning paradigm, we see disruptions in fear-potentiated startle, and we see a non-selective sensitization of the acoustic startle reflex. So these animals do phenocopy to some degree animals which have disruptions in phasic dopamine signaling. Also, we can inject our virus into animals. We can look for restoration of specific D1 receptor signaling by injecting a selective D1 receptor agonist and looking for the induction of an immediate early gene referred to as CFOS. So as you can see here, if we inject this compound into control animals, we see induction of CFOS throughout the brain. And in the animals which lack the D1 receptor, they don't have this induction. We can now selectively reintroduce the ability to induce this gene by reintroducing the D1 receptor through this viral-mediated process. So now we're utilizing this approach to map the postsynaptic targets of dopamine signaling. So what I've shown you is that the NMDA type glutamate receptor plays an important role in regulating phasic dopamine neuron activity, attenuating phasic dopamine neuron signaling, is associated with enhanced fear and anxiety following an aversive outcome. NMDA receptor signaling in dopamine neurons of the ventral tegmental area are the critical neurons important for processing aversive information. And dopamine signaling through the D1 receptor is a likely target for mediating the phasic dopamine-dependent signaling during an aversive experience. So finally, I'd like to thank all of those people who were involved in supporting this work. My former mentor, Dr. Richard Palmer, for allowing me to develop this story and to take it with me and starting my own lab. My technician, Graham Jones, who's been instrumental in establishing our new in vivo electrophysiological system that we're using in the lab. Jonathan Fadock, who I worked with in Richard's lab. Our collaborator, Antonello Bonsi and Emmanuel Argili, who do slice physiology for us. Sherry Mizumori and Minjung Kim in the psychology department here at the UW, who taught me how to do in vivo electrophysiological recordings. Of course, Paul Phillips for his collaboration with Jones Parker in the cyclic voltammetry, as well as other collaborators, Carlos Palladini and James Allen, for teaching me how to make viral constructs. So, thank you. John?
Larry, do you think the same dopamine neurons encode both positive and negative valence information, or are they different sets of neurons?
So
They behave differently when given
the evidence would suggest that they do encode both positive and negative valence. A lot of the original studies by Schultz demonstrating that dopamine neurons are activated. The same dopamine neuron is activated by reward and inhibited by an air puff to the face, for example. So air puff to the face would inhibit the cell, and the reward activated the same cell. So those cells seem to be encoding the valence.
How about for salience in both positive or negative?
So that's a good question. We don't know the answer to that question yet.
You could do that with this.
So we can do that. We can look for activity patterns in these cells, both in response to an appetitive stimulus and to an aversive stimulus, and see if the ones that are activated by both, or whether they're differentially activated. Yeah.
You had mentioned just at the beginning in your introduction that the D2 receptors had high affinity for dopamine, the D1 had low.
Yeah.
Is there any evidence that with repeated exposure to stimuli, like with a shock or other sorts of things, that affinity state changes?
That's a good question. Yeah, I don't know a specific answer to that question, but it's possible that you could have a sensitization or desensitization of the receptor if you have a. So dopamine not only increases phasically, but it also increases tonic levels of dopamine increase with stress. So it's possible that you could have alterations in D2 receptor signaling through desensitization of the receptor, for example, with chronic high levels of dopamine. Yeah.
Can you envision a model using these kinds of approaches where the animal has already learned fearful associations and they can be dismantled with some greater precision with these kinds of interventions? Is there anything where this can turn into a better understanding of therapeutic possibilities?
Right. So what we're going to be doing in the near future is looking for classic treatments for PTSD or anxiety to see if we can reverse the phenotype of those. And if not, can we find drugs that specifically alter phasic dopamine neuron activity? And does restoring phasic dopamine neuron activity to normal level allow you to reverse the phenotype with a re-exposure, for example, which we know to be effective in PTSD patients?
Do these animals have any kind of difference in their extinction behavior? Because if somebody, let's say somebody uses drugs all the time, and the dopamine system has been, for lack of a more technical word, whacked.
Right.
You know, can they benefit from extension training the way other people can for PTSD or humans?
Right. So early evidence in these animals suggests that they have facilitated extinction of appetitive stimuli. So if you train them to lever press for a food reward, they'll extinguish that much faster than a control animal. And the idea there is that a control animal has stamped in that information into much more habitual responding, and the animals that lack phasic dopamine remain somewhat plastic in their ability to change and adapt. so they will actually reverse faster.
So you haven't done that in fear?
I haven't done it with the fear conditioning. We've tried to extinguish cued fear responding in the control animals, and it's pretty resistant to extinction.
But if these guys aren't, then that would be the experiment.
So
work.
that would be, yeah. We also want to look to see if we can, so we can get it to last about 2 weeks right now, the alteration in the intrinsic start reflex. So we want to, and this is a really low amplitude stimulus. So this is 0.2 milliamps for half a second. So this is a very, very mild shock that is eliciting these responses. So if we increase the intensity of the stimulus, can we make these animals basically permanently afraid? Thanks.
Thank you.